We will hear today from Steve Hyman, our wonderful director, and Paola Arlotta, the co-chair of the Department for Stem Cell and Regenerative Biology at Harvard, and also a wonderful contributor to the Stanley Center, specifically with the brain organoids that Sylvia Velasco also works deeply on for the Stanley Center group. And from Bernardo Sabatini, who has his lab over at HMS on the Quad, who is a more recent but very loved member of the Stanley Center community as well. And so we wanted to give an overview of neuroethics, and particularly with a lens on the organoids, because it's very timely. There was just a report out from the National Academies of Sciences that Paola contributed to, that she'll tell us about. And shortly, the ISSCR will be releasing their new guidelines as well. So this is a timely moment, and it gives me great pleasure now to hand over the mic to Steve to give us an introduction.
Thank you, Rosie. I'm going to just give a brief introduction to this whole topic of neuroethics, which is rapidly emerging as a topic of research and discussion, but also becoming increasingly influential with policymakers, with the public, with the press. And it deals with actually quite serious issues, and I'll touch on some of these. But one of them is perhaps something that I personally, and you can see maybe it's a little too flip in the lower right-hand corner of my slide, organoids in a dish, what are they saying to each other? Nonetheless, in all fairness, the public and some policymakers are concerned about, as we start to make human brain models in a dish or transplant human brain cells into the brains of living animals. Right now, we're not allowed to make true chimeras where we mix embryos, and it's also not technologically necessarily feasible, but that would raise even more concerns. I think we do have to recognize that the concerns expressed by the public and by policymakers are important. And in fact, there was just a study of the National Academy of Sciences that Paola participated in as a member of the panel and the study, along with some other people Josh Sains and Rusty Gage. So Paola's going to talk about that, but I'm going to give you just this background introduction. So lots of you already know about basic bioethics before we get to neuroethics. And in less than a minute, the foundations for the bioethical rules that we follow, the so-called common rule, because it's accepted by multiple US government departments and agencies, including the NIH, was founded on something called the Belmont Report, which was written in 1978. And although it came more than 30 years after the Nuremberg trials following World War II, it was a systematization and modernization of the responses that the world had to what was learned in these trials, which included several notorious Nazi doctors who had performed and caused to have performed grisly experiments on humans who were concentration camp prisoners. And of course, ideas like informed consent figured in no way in these experiments, which were painful and often lethal. The Belmont Report defines core principles for the ethical conduct of research on human subjects, and we generally apply it to the selection of experiments, and I'll come down to those in a second, to the assessment of risks and benefits in human subjects research, to the selection of the subjects, and to the provision of good informed consent. So the main organizing principles, the way the Belmont Report responds to these concerns, are to enunciate three principles or values. One is said to be respect for persons, and that's to protect the autonomy of all participants. That means that if you have human subjects research, you must truly inform and gain consent. No deception or lying or manipulation to get somebody to enter your study. Autonomy really means that you provide the subject with the information and the space to decide what they want to do. For example, they can at any time in a human subject's experiment, including a clinical trial, they can decide to exit the trial, and there can be no coercion in that sense. There's a special concern for people who may lack full agency because they're under the control of others, and that would be children, under the control of their parents until the age of 18 legally, military, where people can be commanded to do things, and prisoners. And there are all kinds of special guidance to ensure the ethical conduct of research in the case that one wants to enroll children, because we want to treat children, but people in the military are rarely engaged, but they may be being exposed on the battlefield, say to nerve gas and so forth, that others wouldn't be exposed to, and so they may actually be part of human subjects research. And in the case of prisoners, it was once common to use prisoners in research for every bad reason you can imagine, and now it is very unusual and difficult to engage prisoners in research. The second principle is beneficence, which means that the research study has to be valuable. To warrant any risk to human subjects, the research has to be worth doing. And we interpret that to mean that, for example, that the study has to be adequately powered to give an answer. If you have a human subject study and it is obviously underpowered given the likely effect size before you start, that is prima facie unethical, because you're going to subject people to some level of risk to no purpose. But taking into account the value of the research study and trying to maximize the value of the research study, you still are trying to minimize risks to subjects. There certainly are situations in which if you increase the risk to subjects, you might learn more in your study, but that is precisely what the Belmont principles are worried about, protecting human subjects. And the last is justice, which is that research procedures have to be non-exploitative, and there has to be a fair distribution of risks and benefits to potential research subjects. Now, this is something that has really come to the fore, unfortunately, in the breach during COVID, where disproportionately people of color, people with economically disadvantaged and so forth were much more severely affected, both by morbidity and mortality. This would raise complex issues if you wanted to do recruit people into a vaccine trial in these populations, both because there are higher rates of infections and you get an answer faster, but also because you really have to know whether the vaccine will help these people, minorities and economically disadvantaged people who are being exposed at work every day. You have to really make sure that to convince the way we operationalize this, as you know, is through IRBs, to convince the IRB that you're not simply exploiting an at-risk population because you want to benefit in the speed of your trial from a high infection rate, but that there are potential benefits, not to the individuals in the study, this is research, but to that population, meaning there's an intention, if you're a vaccine manufacturer, to then distribute an effective vaccine to the population that has been part of the study. And I think, as we all well know, in this time when we've been discussing racism and attempts to be anti-racist, that this principle of justice has often been violated, both because we have failed to do research relevant often to minorities and to the economically disadvantaged. But when we do, they often don't then have access to the treatments. And that is clearly an important issue to address. Okay, so neuroethics, of course, takes this standard bioethics as a basis, but it's really concerned with something else, which is it's concerned with mind and brain. And I use the word mind here just to capture, cognition, emotion, lived experience, not because I think that we have some sort of ghost or homunculus sitting in our brains, but mind and brain dependent matters such as personal identity, brain privacy, manipulation of the neural basis of cognition and memory, neural aspects of cognitive and performance enhancement related to the brain, personal autonomy, that is research. This is not just for bringing people into research, respecting their wishes and their agency, but actually doing things to people that might influence their autonomy through drugs or devices. It's concerned with decision-making and the control of behavior. I'll give you some quick examples of this. And then also moral agency and legal culpability. The criminal justice system, of course, cares a lot about whether a crime has occurred and whether this person likely or certainly committed a crime, but is also very interested in the mental state of the person who is accused. And I think we're all aware of the difference between a crime committed in a cold, premeditated manner versus opportunistically and impulsively versus a crime committed through negligence. They have very different penalties, even if they result in the same outcome. And again, we have brain interventions that might influence judgments of both moral agency and culpability. Neuroethics is a field, even the name dates from I think 2002, and it began as a response to new rapidly emerging technologies in human neuroscience. I mean, obviously based often in animal neuroscience, but the focus was on human neuroscience, and increasingly now on the genomics of cognition and behavior as they intersect with neuroscience, and advances in computation, which since 2002 now has a major focus on machine learning. The term neuroethics is unfortunate. It's probably too narrow. And very often people conflate it with the regulation of human subjects research and animal studies, which it isn't. It's really looking at these deeper human issues that are influenced by our experiments and our technology. All right, so here are just some quick examples before I turn it over to Paola and Bernardo. So we develop neurotechnologies, we meaning the research community and the health industry community, to have benefits, typically health benefits, but all of these have potential for misuse in some ways, like every technology, but even nefarious use. So, this is not yet effective. It hasn't worked, but there have been companies that have tried to predict whether somebody is lying from patterns on functional MRI. There was a company called No Lie MRI and another called CFOS that were funded by venture, but also by intelligence agencies, and ultimately the attempt to use fMRI to decide whether somebody was lying was not found to be admissible in court. There's a pretty high bar for what gets admitted in court as science. And these companies went out of business. But the idea that of mind reading using better and better decoding techniques on MRI and using machine learning is something that is not going away, raising questions in the long term about brain privacy. There have also been studies of people with psychopathy trying to predict criminal recidivism, and I think this has quietly gotten a warmer reception from the law enforcement community. Genomic prediction, using things like polygenic scores on an embryo to predict complex traits. This is, I'm talking to a very sophisticated group, and most of you would recognize all of the problems with that, including our limited state of knowledge and the risk of pleiotropic effects, where you think you're selecting one trait, but you're actually selecting other traits. And yet there are now companies open for business. One's called Genomic Prediction, and another was just founded from colleagues at Stanford called Orchid Health, which offers to use polygenic scores to predict things like educational attainment and IQ and height. And there was one in Europe that wanted to predict skin pigmentation. And so you can see that while one might want to perfect the use of polygenic scores and other in pre-implantation diagnosis for severe diseases, very quickly these technologies, once they're approved for any use, get expanded into areas that we might think really represent a very poor idea. I'm not going to go through the whole list because I don't want to take too much time, but there are 200,000 people in the world with deep brain stimulation electrodes, mostly to treat movement disorders, but there's a lot of experimentation to try to treat major depression and OCD with deep brain stimulation. Clinical trials data is not all that positive yet. Interestingly, much of this research in the United States is funded by DARPA, which the Defense Advanced Research Projects Agency, and sotto voce, but not so hidden, they mention their great interest in enhancing soldiers, or as they call them, war fighters, or connecting the brains of soldiers to computers and robots, because after all, humans are good at some things and computers are good at other things. I will skip the rest of these because I think you get the feeling as why this consideration of functions of mind and brain have become a matter of increasingly, I think, good deliberation and discussion. Now, why does this matter for brain organoids? Well, one aspect of let's call it neuroethics, which is very much grounded on our self-awareness, often not only cognition, but metacognition, emotion, the ability to feel pain, and so forth, is the question of moral status. And moral status matters because if obviously humans have full moral status, but moral status is something that is not black and white, yes or no, but rather it's dimensional. And we consider animals to have some level of moral status. This is not always stated explicitly, but it's why we care more about non-human primates than about invertebrate animals, for example. The typical definition is that an animal can be said to have moral status if its suffering is morally bad on account of the animal itself, regardless of the consequences for other beings. That is, we might say it's bad if an animal is feeling excessive pain, it'll mess up our experiment. But the idea is that we have attributed to this animal a status in which its suffering matters just in itself on account of the animal. And this really underlies a lot of the law and regulation and moral guidance. So this is the question that's now been raised about human brain models. Let me just get this back. about human brain models. The question is, not today, but can we in the near future make brain organoids? I'm sure Paola can do anything actually, so complex anatomically and functionally that we must consider according them some level of moral status, and then we'll have to regulate them and use them accordingly. The same is true of how many cells and what kind of networks transplanted of human cells transplanted into an animal brain might have that kind of effect. What would happen with true chimeras if we could make them when we were allowed to make them? But in these cases, often what's lost, this is why it's really important to hear not only from Paola, but also from Bernardo, people are worried about, and I think we have to take these concerns seriously because after all, we want to retain our ability to regulate ourselves by doing what is perceived to be and is the right thing. People worry that an organoid might be sentient and in some way that we don't understand, might be experiencing suffering or distress. But the antecedent science question is, how would we even know? What criteria would we use? How would we make this difficult inference? Now, I'm going to stop there, but it hasn't stopped some scientific colleagues who I think should know better from doing some recordings from organoids and saying this is organized brain activity and maybe they are conscious. And with that, very quick primer on neuroethics and how it's being now applied to human brain models. I think I'm turning it over first to Paola, is that right? Yeah. Okay. So I will unshare so you can share.
Yeah, thank you, Steve. Here we are. Here we go. So this is, of course, an extremely important topic. And with science advancing at the speed with which science in this field, not only that of neural brain organoids, but also that of transplants and human animal chimeras, it requires a deep conversation about the ethics of these systems. And this conversation can happen in many shapes and forms, but I feel that the one that I'm about to present to you was perhaps, at least in my opinion, one of the most meaningful type of conversations on these topics, because it was set up in the form of a committee that I'm about to introduce in a second, but it really spanned a lot of time, meaning almost, was something like seven or 8 months of very frequent meetings where we listened to a lot of experts in a lot of different fields and processed a lot of information, followed by deliberation and brainstorming and really thinking about this topic deeply. This committee, so this work was commissioned by the National Academy of Sciences, was sponsored by NIH and the Dana Foundation. And the co-chairs of the committee were Bernard Lowe from UCSF and Joshua Sains from Harvard University. This was the committee, as you can see, was composed not only by scientists. It shouldn't be only made by scientists, of course. It was also made up of lawyers and ethicists and religious scholars and of course scientists. And the kind of speakers that we invited to speak to us also reflected a very broad areas, very broad fields, both in ethical space, in scientific space, religious space, and so on and so forth. So there was really sort of a deep discussion, perhaps among people that normally wouldn't necessarily talk to each other as scholars, or at least not on these topics. And it was really, really nice. So first of all, how do we start this, and why did we do this? I mean, this is not the audience that I need to explain this to, but it was really motivated by the fact that neurological and psychiatric disorders are, of course, major causes of morbidity worldwide, and so are neurodegenerative diseases of the kinds that I'm describing here. And that progress in understanding brain function and dysfunction, it's really increased in the past decades, but treatments have lagged behind. And there is sort of an appreciation for the fact that preclinical models, for example, rodents, while very important, they also have been shown to fail at exhibiting key features of some of these complex human diseases. And studies, on the other hand, on human subjects are quite limited, of course, for technical, legal, and ethical reasons. And so additional models are needed, and these are the models shown here: neural organoids, cell transplant, and neural chimeras that we need to discuss as a community. Of course, with the promise of these human experimental models of the brain that we listed come concerns, and some are common to many new technologies and areas of science, but others are kind of unique to the special nature of the human brain, which really defines us as individuals. And perhaps the best way to show this, if you think about a transplant, if your kidney was transplanted into me, I'll still be me. But if your brain was transplanted into me, paradoxically, who would I be? So I think that this question really sort of exemplifies why we talk about this kind of things. Now, most people feel differently about human neural organoid, transplant, and chimeras than they do when similar technologies are applied to non-neural tissues, and we can all agree on this. Now, what was the charge of this committee? We were not charged to make recommendation by the end of our deliberation, but we were charged with searching for information and for processing sort of this information with all our very different backgrounds and expertise, and produce a document that would then help other agencies to think about potential regulation and so on and so forth. Questions that we included when we were doing our research were things like: how would researchers define or identify enhanced or human awareness in a chimeric animal? Do research animals with enhanced capabilities require different treatment compared to other and more sort of typical animal models? Here is the difficult one: how large or complex would brain organoids need to be to worry about enhanced or human-specific awareness capabilities? And also legal questions: should specific consent be required for patient cells, for the use of patient cells to create neural organoids, for example? And are there gaps in current regulatory frameworks relating to these systems that we should consider? We report findings, we don't make recommendations. We all know here what organoids are, but just very quickly, these are three-dimensional systems that can be derived from pluripotent stem cells, either induced pluripotent stem cells or embryonic stem cells. They come in many different flavors, but fundamentally can be generated either largely by self-organization, self-organization, so unguided differentiation. And this typically leads to organoids that may represent multiple regions of the brain. They are highly, I would say, unreliable and not reproducible in their development, so you don't always get the same type of organoid from this system. Or by guided differentiation, whereby the investigator imposes signals during differentiation such that you can make specific or bias the differentiation of specific brain regions, which themselves can in some cases be fused to form what are called as assembloids. Now, organoids generated from patient-derived stem cells can in theory exhibit disease features, and we have talked about this extensively here at the Stanley, and can be used perhaps to identify mechanisms and in the future to inform potential therapies. Now, they are currently limited in the size and complexity that these organoids reach, And they're quite simple if we think about them and compare them to human brain, and we think about what the function of the human brain really is. The other two systems that we discussed that were actually much more complex than the human organoids for their ethical, human brain organoids for ethical discussion were the transplant and the chimeras. And here I have to tell you exactly how we defined, especially the early blastocyst chimeras versus the transplant, because this was a matter of definition of how we call this system. So in this report and in our deliberation, we consider transplants related to the brain, situation in which human cells derived from, for example, from human pluripotent stem cells, they could be grown in monolayers or in organoids or in anything else, are transplanted specifically within the nervous system of another species. Here could be a mouse pup or an adult mouse, but you can also think about transplantation in a non-human primate in a non-human primate brain, so in the nervous system of an animal, versus a chimera that is generated by mixing embryonic stem cells within the blastocyst before gastrulation. And the difference that we wanted to make here is that in this case, in the first case, this type of transplantation tends to result in chimerism within the nervous system itself. And it's very, very, very unlikely, in fact, considered impossible, basically, or very, very unlikely, I would say, that these cells would contribute to germline transmission. When in this case, the cells have the potential to colonize every organ of that chimeric animal. So it's a more extensive, potentially kind of chimera, and can also colonize the germline. So we analyzed a lot of data, we listened to a lot of experts, we read a lot of papers. And here are some of the key scientific findings, just to get us to the bottom of what we found. By the end of this presentation, I'm going to give you a link to the actual full report, which is quite consistent if you want to read the details. So from a lot of these talks, it emerged that animal models are important, but in the context of modeling human brain disease are inadequate. And the research on human is limited, of course, by legal, ethical, and technical issues. And this poses a problem that made the majority of the scientists and ethicists that we interviewed conclude that human neural organoids and transplants are needed, and they're needed to circumvent some of these limitations, enabling new approaches to analyze human brain development, to discover mechanism of human disease, and eventually to test potential therapies. So it was considered that this system would offer the opportunity to go in that direction. Now, we also found that now and for the foreseeable future, and by that, nobody can say when that foreseeable future will end, but now and for the immediate future, it is extremely unlikely that organoids would possess consciousness or emotion or experience pain. So we got to writing this sentence again after a lot of thinking about the nature of the systems and what would be needed for us to consider this a problem. So there was a general agreement, certainly among all of the members of the committee, but also among all of the scholars that we interviewed. Nobody seemed to be particularly worried about organoids today. However, there is this thing of the foreseeable future. We have to continue to think about this. To date, it's not possible to generate these viable human neural chimeras in any non-human species. So there are technical and biological limitations to this, and this was one of the conclusions. However, the committee identified and analyzed several concerns that the science related to these systems really raised, and these were ethical issues, current oversight and regulation, and we also identify as an area of intervention the value of public engagement and communications. And so the ethical findings specifically were that there are strong moral arguments in favor of human neural organoids, cell transplant, and chimeric research. which is directed at studies that would relieve human suffering, as long as the research is balanced with ethical considerations. So the kind of discussions and research that we did needs to continue and not just be a one-time phenomenon, because these are systems that do require ethical considerations. It is extremely unlikely, as I mentioned before, that organoids would possess today, in their current form, would possess awareness, consciousness, emotions, or experience pain. And we think that this is not a concern that would sort of change in its nature in the immediate future. And there was wide agreement that animals may be used for research directed towards the goal of relieving human suffering, provided that the research is justified, as Steve was mentioning, harm to animals is minimized, and the needs of the animals are met. However, several issues emerge also. There are diverse views based on religious and secular values, which must be respected in a society that is pluralistic. There was the issue that emerged many times of blurring the distinctions between humans and other animals. The revulsion at threat to human dignity, basically, and the possibility that animal may acquire human capabilities and behaviors that resemble human behaviors. And there was a recognition of the special status of the human brain as determinant of human identity. And so every time a chimera is generated, especially affecting the nervous system, these considerations emerge and they need to be met. There were concerns about animal welfare and animal rights as the recipient of these transplants or as these chimeric animals who could acquire enhanced capabilities such as experience of pain and consciousness, greater concerns about non-human primates as hosts. So there was a distinction in the discussion between who the recipient, the animal host would be. So discussion become much more complex when it's a non-human primate versus a rodent, for example, because of the different moral status. And as models of human disease improve, also may show behavior that would be considered distressing in humans. So these were the ethical findings from the committee. There were also considerations related to consent that we talked about. There were arguments both pro and against additional consent requirements. For example, some donors may not want their biological material to be used for projects like the ones I just described, and they would object to it if they knew about it. There are concerns about re-identification of biospecimens after identifiers are removed, and so it's possible to go back to who the donor was. And we also need to be aware of past ethical violations that were pretty prominent during research with African American and Native American participants. And this makes this topic particularly sensitive for these populations and others. There were also arguments that we heard against additional consent requirements. A consideration like the fact that people should not contribute biomaterials they have given away or biomaterials that they have donated or abandoned. And then additional consent requirements for existing materials could prevent further use of the difficult to obtain material. So the more consent requirements we layer on the use of the cells for these purposes, the more we slow down research, and some sort of an equilibrium has to be achieved there. So at present and in the near future again, ethical concerns on neural organoids, the transplant and the chimera are addressing the context of oversight mechanisms like escrow or IRB or IACUCs at all of the institutions where we work. And it was recognized that these committees often lack the expertise to really judge the type of work that involves neural organoids and chimeras. And so there was some concern that emerged from the discussion about that, and the need to assess oversight as science develops. And it was concluded that neural organoids will not raise issues that require, at this moment at least, additional oversight, unless they become significantly more complex than what they are today. And so I have a couple of additional slides, and then I can pass it along to Bernardo before we open to the discussion. So it was also concluded that future research will benefit for a continued conversation on the ethical and social implications of this work. And everybody recognized that there are advantages to holding this discussion at the national level. At the moment, there is no national body in the United States that addresses this need. The science is relatively new, and the concerns are relatively, at least some of them in some of these systems, certainly from brain organoids, are new. There is nobody in the United States that does this, although there are some committee within NIH that may provide at least an entry point for this, like next track. In several areas of biomedical research, it was mentioned, there are oftentimes there is the use of the traffic light system, whereby different experiments and different type of research gets classified as green, yellow, or red, depending on what the state of the science is, whether it can proceed without oversight, whether it requires some heightened oversight, or whether it cannot be carried out at this moment. And there was some suggestion to adopt this system in the context of organoids and chimeras. Public engagement and communication, there was a lot of talk about how we as scientists communicate this science to the public, and how oftentimes, and especially in the popular press, the models and systems that we describe here are not described correctly. Oftentimes to be sensationalist about the system themselves, but this really informs public opinion the wrong way. And everybody recognized the need for greater public engagement in this discussion, but that requires mechanisms for engaging the public. It requires mechanisms for making sure that that public is a representation actually of the different religious and secular views of the society. We need to start engagement in a more meaningful way that also empowers the scientist to provide information about the system that can inform a valid discussion. This is an example for how oftentimes the opinion of the society on certain type of work is so much influenced by the way the scientists describe the work. For many, many years, the word mini brains was used every time a human neural organoid would be described in the press. And that's just wrong, because this evokes a vision for, for what this system are that may really compare them and really equate them to the brain itself, which is not what the system are. And therefore the conversation then it's deviated in directions that it shouldn't go into. And so we need to pay closer attention to how we describe this system to inform this discussion. So let me summarize a little bit. I know there was a lot of information here, but it was also many, many months of work and discussion. Given the devastating toll of brain diseases, the public has a strong interest in research advances that offer hope for the treatment and cure. And human neural organoids, these transplant systems and the chimeras, are important models for this type of research and can provide important insights into the functioning of the human brain and the malfunction in the context of disease. And as the power of this research model advances, we need to have more and more ethical discussions about them, because the ethical concern will for sure rise. These ethical concerns need to be addressed, while also allowing justified research to actually proceed with care. Existing regulatory oversight system address this research at present, but periodic re-examination will be important because this science and these models will evolve, and it may evolve actually rather rapidly. I am going to stop here. If you go at this link here on this webpage, you'll find the full report. It's organized quite nicely with the science first, and then all the ethical considerations, as well as the legal consideration on the system. So if you're interested, there is a lot more information there. With that, I'm going to stop sharing my screen.
Yep.
And
but Bernardo, yeah. So Paola, that was really great. That was a really lucid description of a complicated report. But you know, one of the concerns that the public, and again, our policymakers might have is, while I totally agree that we don't think that these current organoids are sentient or suffering, people might ask how we know. And what are you going to do in the future to measure?
Yeah.
And I don't want to put, it's unfair to put Bernardo on the spot because it's very hard to interview an organoid, but
I can tell you about my thought process, obviously.
Yeah. Over to you, Bernardo.
Yeah, I mean, I have no answer to your question, but Paola, do you want me to start or do you want to?
Yeah, please.
Yeah, go ahead. And then I can tell you what we discussed about that.
Okay, great. Yeah, so I'll just start off by saying, look, I'm not an expert in this topic, as Steve and Paola know. And I'm not going to give you an answer, but rather I'm going to tell you a bit what was the thought process, you know, in my household over the last week as we debated this kind of vigorously, and then sort of what, as an experimental neuroscientist, I think may actually be a tractable way forward. So, you know, the question is this then, you know, is an organoid sentient? And Steve, you know, talked about sort of the neuroethics perspective and how we need to think about the presentation of our science to the public and the worries of the public, you know, very well, I think, justified worries, even if we're not at a stage yet where any organoid is sentient. There's another way to think about it, though, which is sort of as a biological goal. So, you know, one could imagine that somebody comes to us and says, look, I have figured out how to grow 10,000 organoids at the same time, and I'm going to put them in different conditions, and I'm going to get a sentient organoid. That's my goal, right? And we have to figure out how that's done. And in fact, as was mentioned earlier, Alison Motri at UCSD explicitly says that he wants to make a conscious organoid because he wants to study autism, and that the only way the organoid will be useful is if the organoid can exhibit autism, right? And so the question then, of course, is how can we tell if he has succeeded? Of course, he hasn't, you know, but how do we know that? And I think that, you know, we can address this question from a variety of different perspectives. And I think actually the people that we should talk to the most are those in artificial intelligence working on sentient AI. And I'll explain to you why in a moment. But you know, there are a few different camps in this question of sort of sentience of organisms or of computer programs. And I think the AI is the one that's most useful to us. So the first question we struggle a lot in the household is, you know, sort of, does the body matter? And this is something that's heavily embedded in this question of sentience. And I think the answer is no. And just forgive me for giving you a couple of very simple examples, right? So, you know, you can imagine that there's a person that has a, you know, yet an unheard of disease in which all non-brain tissue dies away, right? And in order to save this person, we take that brain and we put it in a jar and we keep it alive. And so, you know, is that brain devoid of a body and of any sensory system or motor system conscious? And I think probably most of us would say yes, you know, and we would imagine this is quite a horrific fate because we imagine that brain as conscious. And then on the other extreme, you know, if we take an oyster or whatever organism you might think is not conscious, you know, and endow it with a sophisticated life support system and an exoskeleton and a face and allow it to wander around Newbury Street, you know, would it be conscious? And I think most of us would say no, you know, it's still an oyster, right? And so I think most of us would agree that this question of sort of sentience or consciousness is really intrinsic to, you know, the organism, organoid, or computer program that we're talking about, whether or not we hook it up with the proper sort of body-like features. So I think with that in mind, we should say, okay, well, form doesn't matter that much. And it really is valid to ask if an organoid is conscious or sentient, even as it's sitting in a dish. And so, you know, for me as a non-specialist, what I've struggled with, and I think is what the whole field struggles with, is that we don't really know how to define consciousness or sentience, right? And we know it's like Edward Meese said about pornography. You know it when you see it, and you know we have a feeling for consciousness. We kind of understand what it is based on our own, you know, our own self-awareness. But it's very hard to define. And so, you know, there's a few different levels of definitions that I've found in literature. So there are some people that go all the way, apparently, to a tradition from William James, although I haven't followed it back in literature, go all the way to say that, you know, life is by definition sentience. And these are really the same thing. And they propose something that they call the cellular basis of consciousness, arguing that every cell, because it can react to its environment, because it can change in response to its environment, because future reactions are different from previous reactions because of these changes, that each cell is conscious. Then I'm going to focus mostly here on this intermediate level. Eva Jablonski has attempted to really define behavioral sort of motifs and tests that you can do to an animal to define whether it's conscious, and I'll spend a little bit of time on that. And then there's, of course, this long tradition that I'm sure Steve can speak to at great length that comes from, you know, psychology and from philosophy, which is sort of around qualias, right? And so, you know, the qualia is the feeling or of having experienced something. So, of course, many organisms can react to a noxious stimulus, right? But only some organisms will really experience that as pain, have the qualia of pain. And so some people will define sentience as having that qualia. I think that's kind of a low definition. Most that I can read about will define it instead of saying not only experiencing the qualia of pain, but being aware of your own sensation and qualia of pain, such that you can recall it later, right? And so this is really an important concept because it says not only do you react to pain, but really you internalize and you have an introverted sort of access to that experience that you can relive. So I think the first and the third here are really not very useful. The first one is one that maybe I will naively but sort of discount kind of out of hand. That's sort of inconsistent with our sort of belief as to what consciousness is. And then the third one I think is just not useful, right? When we're talking about an organoid, there's really no way I think to probe this question of qualia and access to qualia. So actually I preloaded, if I can find it. Well, I'll have to do it later because I can't do it while I'm sharing screens, but I'll post this link in the chat after. So there's a Frontiers article in which this researcher, I don't know her beyond having read this, Eva Jablonka, attempts to define what it means to be minimally conscious, right? So minimal consciousness. And she's really doing this thinking about animals, okay? And I think it's a useful exercise, and she tries to do it from sort of an evolutionary perspective, sort of look through evolution and say, well, what are the features that an animal has that makes it be minimally conscious? And they're listed here. I'm not going to go through them, but they do involve these sort of ideas of there's like an internal state and sort of binding problems and things of this sort. And the problem with this list, and I'd say the problem with all of the debates about consciousness in animals and lower animals, is that they really come from this animal perspective. And so they're always phrased with the concept of a body and of self-driven survival. Motivation to survive are always implicit in these kinds of animal perspectives about consciousness. And of course that doesn't apply to an organoid, right? It'd be very, very hard to say that the organoid in the dish has a self-driven survival to survive, right? Self-driven survival instinct. But what I do like about this article is that she tries to define behaviorally measurable features of an animal that should be there if they have those properties of minimal consciousness that I showed you in the previous slide. And so she defines something that she calls universal associative learning. And we may not like the details of her ideas, but I sort of applaud the approach that she's taken. And in this article, she defines what that means, universally associative learning. It has some very interesting features. By her categorization, aplesia and C. elegans fail. Okay, so they cannot do universal associative learning. One of the key things for her is this issue of sort of complete versus elemental learning. right? So you can, for example, teach a very low organism that red is bad, but it's very hard to teach it that a red square in this context in the following way is bad. So the binding of all of the sensory features of the environment often fail. You can teach a relatively low animal that red is bad, that bright lights is bad, but you can't teach it that red in a dim situation is good, right? So it can't combine elements at will. So this is one feature. But what I like, though, and this is sort of schematized on the right, is that she then goes on to say, well, what are the features of a nervous system from a structural point of view that would be necessary to support universal associative learning? So here she's starting to get to things that one can measure from stimulus response properties, and then that one might be able to probe sort of anatomically. So this then suggests that sort of activity and structure can be used as evidence of sentience. And I think that this is something that the community reacts to and believes. For example, when these organoids were reported to have developed fetal-like EEG patterns, people had sort of a shudder. It doesn't seem really to have been true, right? But when Seston, revived pig brains, after death, and some of them developed EEGs, the immediate response was, oh, let's anesthetize them, right? There was some trouble there because of the electrical patterns. And certainly, if Paola managed to grow an entire human brain in a dish, we would be troubled, right? So the way I think we need to move forward is that I like to phrase this as a practical question, not a philosophical one. I think it's not helpful to get into philosophical debates about what it means to be sentient and to talk about, absent qualia and zombies and this kind of thing, but rather to remember that we have a practical question. How do we determine if this organoid here in a dish is sentient or not? And that's why I think we really need to engage with researchers in the field of sentient AI, because they're also dealing with a problem in which they have a device that's separated from a body, okay? And the question is, how do you probe that device, which doesn't have an animal origin, and ask if it's sentient? So I think, if we really want to go down this road as an internal exercise, we could start to define the features of organoids, sorry, that's an autocorrect there, of organoids in the following categories that would trouble us. So the first one, Paola alluded to, which is sort of structural motifs and levels of complexity, right? Now, this isn't very complicated because I looked up last night, what's the smallest mammal? And it's this thing called an Etruscan shrew, which has apparently a 60 milligram brain, okay? And yet it can do most of the things that simple mammals can do. So size really isn't enough. Then we have to start defining, input-output relationships, right? So what can we do to test this organoid to see, can it actually form something like a memory? Can it do universal associative learning despite not having any output or input to the outside environment? Of course, Paola has sequenced organoids in which there seem to be things like photoreceptors there. So of course, once you start saying that there might be sensory organs, then you can start to ask this, I think, in a more profound way. And the third one, of course, comes around patterns of activity, right? So maybe there are features of EEG-like activity that would bother us, maybe not. But I think most importantly, what are the kinds of features that you could look at that would suggest that an organoid has kind of an internal state, right? Or that internal state can be changed and endow it with a memory after some form of associative learning. So that's all I have. So again, no answers, but I do think that in maybe engaging the field of sentient AI and trying to think about at least these three types of questions that are here would be fruitful, and continuing to leave it on in a practical domain as opposed to a philosophical.
I mean, that's a brilliant discussion. And I think the focus on practical experimental questions is the only way forward. Paola, I'm wondering whether you have any comments or reflections, especially given that you early on developed organoids that responded to light, that had retinal cells and had a physiological response.
Yeah, absolutely. And thank you, Bernardo. This is fantastic. These are exactly the kind of things that we discussed quite extensively. Speaker after speaker in the field of consciousness broadly interpreted, nobody could, it's like you say, nobody could really give us some indication of what the neurobiological substrate of consciousness would really be. And when would you start getting, where are the boundaries? When would you start getting worried? When would you not be worried exactly, and so on and so forth. So certain things emerge from this discussion. For example, the need for a certain level of complexity, but also intended as an experimental, let's say, brain organoid model that would contain multiple brain regions. So recognizing that not only one brain region may be important for this, but that actually connectivity, especially long distance connectivity of multiple brain regions, is very important. But you can argue that by creating assembloids and things like that, perhaps even in the near future, you could achieve that. The second is this idea of having sensory input and experience, right? In a way, let's say sensory input first. As you can say, I have photoreceptor, I can shine light, and you can see an effect on the level of activity of the circuit within the organoid. But then you need to ask yourself, what is that beyond sort of the light stimulation of a photoreceptor, which is a cell built to respond to light. The discussion went a lot into experience and the fact that you sort of need past experience, past experience in order to generate a conscious state of a certain type, and that organoids don't collect that, at least in the current conditions. You can imagine a lot of different scenarios in which some primitive forms of this could be imparted to organoids, but the lack of experience, the lack of complex long distance connectivity, the lack of coordinated input of sensory input was recognized as some of the major limitations to perhaps establish some sort of probability that this system would have consciousness. But it's difficult.
Bernardo, do you want to? There's a question on this topic from Stuart. Do you want to read it out loud, or should I?
Yeah, and Stuart, if you want to ask your question directly,
yeah. Stuart could read it out loud. Yeah.
I hesitate to because I am just an interloper being.
No, you're Stuart. Look, none of us know. I mean, I thought what was really great about Bernardo's discussion was his humility in going to what I think actually is the best paper and describing it in that context. So don't feel inhibited.
Well, yeah, no, and I totally agree. I was incredibly stimulated, Bernardo, by what you had to say. And I very much like getting at a definition that is somehow measurable. Otherwise, this is just a philosophical discussion and we can go round and round and round. And but it felt like the bar was too low. That for me personally, it was just a visceral response. And I wrote, if an organoid could learn to distinguish red from blue, I have no problem. That doesn't bother me. And here's where language is so complicated here, but if it could show somehow a self-awareness, in some way, you had evidence that it knew it was an organoid.
Metacognition would be the
metacognition.
Term.
Yeah. Evidence of its existence. Then I'm worried. Then I'm really worried.
Oh, we'd be terrified.
And of course, I worry this about my machines also. If my machine says to me, please, Stuart, I'm not feeling secure. Don't turn me off right now. I'm going to freak out, right? If my machine can figure out all kinds of solutions to my science problems, so be it. So, anyway, that's
no, but so what you're saying though is raise the bar, but this has to stay a pragmatic and experimental problem. I think what we have to remember is that we may, partly because of scientists who unfortunately sensationalize, but also because people really can be worried about these things. We may face regulation unless we can describe the case, right? A consensus threshold for being concerned.
Yeah, I agree with that. And I agree also, by the way, it's pretty obvious that my statement does not provide that. I'm struggling with the words and maybe even the concepts. It's easier for me to say what I don't consider as evidence of, consciousness, as opposed to, a processor. Like a machine is a processor, but it's not conscious. So it's easier to say what doesn't worry me or what is not, but it's harder to put into words or define the measurable that is evidence of consciousness.
We have a contribution from Dr. Lunshoff. I don't know whether.
Please unmute yourself and contribute to the conversation, Dr. Lunshoff.
Yes, hi. Yeah, I'm happy to do that. So I'm a philosopher ethicist and
Great.
Working in neuroethics. And Steve, you may know I've been in the neuroethics working group, and Paola participated in our brainstorm project.
Argentina.
So it's I think it's maybe amazing to many of the scientists listening to be aware that philosophers and ethicists actually set the bar much higher often than scientists do, and then of course the lay public would do, because it's really important, and it's a big and hard to answer question whether being sentient as such is attributes like moral status to a living being. And that's, by the way, one of the problems with AI, like is AI alive or not alive? I would think it's not biologically alive, but even biological life, does that have moral status, or is it, as we tend to say, like morally considerable? Does it have interests that are relevant to others? Having interest of yourself and for yourself that are so important that deserves protection, like you're keeping those interests, you're not getting harmed. That's a very, very high bar. And like, in my opinion, but I'm a consciousness skeptic, and for sure not, many of my colleagues share this view, like my good colleague in Soyun, for example, we have the same views here, but many others, of course, do not share this opinion. We do not think that brain organoids meet this criterion.
At least not now, yeah.
And of course, the problem, the fundamental problem is if you cannot define clearly, unambiguously what consciousness is, how are you ever going to measure it? And
well, that's the
crux of the
that's the crux.
Exactly.
That is the crux. Yeah. But again, we need to have these kinds of discussions and even be able to write down some conclusions, because I do think that at least in some places, regulators will get squeamish and will impede important science, whether it's organoids or xenotransplantation of cells into animals. And again, right now, chimeras are technologically impossible and also forbidden by NIH, but it doesn't mean that in the future that won't change. Stuart, if you want to say something else on this topic, go ahead, because I was going to ask everybody another question.
Really, really simple. Of course, none of us know the answer to this, but I agree that it doesn't seem to me to make the distinction of biological or abiological doesn't seem helpful, because, I do imagine somewhere out there's sort of abiological intelligence and consciousness, and it just seems likely to me that the biological consciousness is like the beginning of building machines that have consciousness, and they're going to probably outlast us. But so it just seems to me not so useful. Of course, there's no evidence for what I just said. Maybe I'm wrong. Maybe it's just something totally special.
No, Stuart, if you're not a mystic, biology is one way of implementing, these brain functions, but it doesn't have to be the only way.
Yeah. That's all I'm just saying is.
Yeah, no. So I was going to ask another question. So we don't have that much time. We consent people to make stem cell lines. We don't, we might in the consent say vaguely, that they will be used to study human disease or human neuropsychiatric disease. But we certainly don't say in the informed consents that we may, take your cells and make in vitro an organoid, which is a, small and incomplete and somewhat disorganized, but a replica of your brain. Is anyone troubled by that? I would just open this up. If not, I can always turn to Paola, who's probably thought about this.
Because we asked every person that came to speak to us, and also we really wanted to have multiple expert of multiple religions and different cultures and so on and so forth. And we were a bit surprised perhaps, but nobody voiced concern about these organoids. And this question was asked as well, and the conclusion that I listed, that at the moment, there doesn't seem to me to be a need for additional consent because of the nature of the organoids today. But I would also say that it's just a moment in time, and maybe tomorrow, as these things become more worrisome, if they become more worrisome, there may be a need for that. It's just that it was not necessarily, perhaps unexpectedly so, was not voiced as a concern. In all of our discussions. It could have been that many scholars with some of those specific ones, but.
Yeah, I mean, there's a comment from Shana Malat, who is, I see Rosie's answer, who would like to make a contribution, and this would be a great way to do it without having to donate the brain. But of course, not everybody would necessarily agree with that, and I do think it's a right now. I think we feel that if we went into the detail of everything that the stem cell lines would be used for, we would just dissuade people. But later, we might actually be using stem cells and organoids that are re-identifiable as that person's in order to screen for personalized medicines, which is now what's done in cancer biology when people make colon cancer organoids. I think Stuart, right? Isn't that true? That you want to reconnect it to the person. So we're not there yet.
But if I'm saying, I'm Shana, if I'm saying that I agree to that, that is what I want. I don't care about being identified. And if there are other people like me, why does it matter if the public is maybe not okay with that? That's part of me. And I'm saying, yes, please do that. We can't expect non-scientists to understand that.
So to give you a quick and inadequate answer is that an institutional review board giving permission to consent people to donate cells, right? Once the person has donated cells, the IRB goes away, but they may actually be concerned about, the IRBs tend to be very, very concerned about re-identification just for the last several years, as opposed to what they might alternatively be concerned about, which is what is done with the information if somebody is re-identified. Is it a positive and benign use that the person might want, like using it to screen for their version of an illness? Or is it for some inappropriate, intrusive marketing or something? It's a real frontier.
I think that Shayana also raised a very, an additional point that is really, really well taken. And this was part of the discussion too, of whether it might be actually unethical not to use these models if we think that they have the potential to change the status quo of science, right? And understanding of disease and development of treatments. And so if we make this science too repulsive because of whatever reason, then it might be actually unethical to do so, considering. So everything has to be put on the same plate so that we make our ethical consideration also in the context of what the potential that this system have for new discovery and development of treatment and new opportunity, even if today we cannot, materialize all of that potential, but it might be unethical not to make it continue to progress.
Exactly.
And so, yeah.
Just because, living with it for a decade, and it's when you look at how much people don't research depression, understandably, because there's just so many aspects of it that we can't really manipulate in an animal model, and it's so subjective in humans, that we need to understand the brain more. So it's frustrating for 10 years pretty much to be told we have nothing new, we have no answers. I would rather something be done, just something. So yeah, it's frustrating when you have this roadblock. It seems unethical to not try to take that step and just say, well, we don't know.
Just completely open. Any questions or comments? If not, yeah, go ahead.
I was posing one more question. So what do people think about neural chimeras intended as the transplanted, let's make it particularly sort of difficult perhaps. How about chimeric animals that where a non-human primate brain receives human cells generated perhaps within an organoid or a 2D culture or whatever else, and either during sort of the development of the brain, which would be the time when these cells, transplanted cells, would have a greater opportunity for integration and form these low level, but yet chimeras, or postnatally where these opportunities are a little bit more reduced. I wonder what people think about that kind of experiment.
Paola, would there be safeguards that could be built into those neurons as a kind of suicide gene for the human neurons, so that if the animal started behaving in a certain way, you could cut off all the human neurons in the brain?
I mean, one can think about all sorts of safety nets, but the sort of the question still stands of whether this type of experiment should or should not be done. And maybe I'll add a little bit more color, because from a scientific viewpoint, that specific kind of experiment would provide answers that no other system at the moment could. So there is a limit to what can be studied in an in vitro system. For as complex as an organoid can be, there is a limit. And there will be things that you can study in vivo that you cannot study in an organoid. And there will be things that you can and others you cannot study just in the animal, in mouse or in a non-human primate. So these chimeric animals may actually be extremely powerful models to model, for example, reaction to to reaction, sorry, pick up in school. So reaction to an environment or experience or right, that you couldn't test necessarily in vitro. Yeah, that can tell you a lot about the cells and the context of disease and how they would react and so on and so forth.
No matter what organoid treats you put into your bioreactor, it's different than growing up in a circuit in a brain.
Right. I mean, my personal take, and others please pipe up, is that I personally would be fine with it if the animal was not in pain. And I remember Guoping saying at one point that people had asked him whether he would model certain childhood diseases of epilepsy in non-human primates. And he said he just couldn't bear the thought of it, because he just couldn't bear the thought of seeing those animals fitting so constantly, you know? And so I think if there were things that we could study in the animal that wouldn't cause them pain, I would be okay with it. That would be my bar. I see we have a hand up from Nathan.
So my understanding of this is probably fairly limited, but if you're going through with making the chimeras, there's the transplant of human cells, and to an extent, they're the iPSCs that could also go forward to make organoids, which are, for a better or worse kind of context, they exist as kind of information processors, as opposed to something with enough, say, critical mass to have a consciousness or be maybe like the low level brain that really kind of evokes a more visceral reaction from kind of people in general. So if the chimeras only exist with, say, a specific animal, per se a mouse, and then the subset of cells that lack kind of, again, the critical mass to develop what we consider consciousness, then if both of them are considered to be kind of subconscious is a poor term for it, but I think you understand where I'm going with it. Perhaps the integration of the two will create some sort of thing that per se models consciousness, but without actually doing it, without knowing whether or not you have that reaction, I don't see why it would be necessary to consider the ethical implications that are associated with consciousness, or why the chimera would have to be classified differently as a mouse model experiment or an organoid model experiment.
I guess most of the or some of the concern may come from the fact that now you are integrating human cells with human properties within a circuit of another species, and you may potentially alter the functionality of the host circuitry. Now, to what extent this would be altered, what kind of output you would get, would it be pain, as Rosie was describing? I wasn't even thinking about consciousness for the moment, more like enhance functionality of that circuit, or whichever way you want to define it. Would that be troubling? Because the sort of positive, in a way, implication of their work are very clear. This would not be done just for the sake of it. This, in my opinion, would be done because it would offer an opportunity to study something that we couldn't otherwise study, that may be really relevant to understanding disease, but it's more charged. Yep.
Cameron Pernia mentions, we know the Steve Goldman experiments transplanting glia that presumably, according to his findings, improved some of the cognitive capacities of these mice. I've never known what to make of those experiments, but people have also transplanted neurons. I mean, people are making these xenotransplantation chimeras, but they just haven't made them with large numbers of cells in non-human primates, which was Paola's really hard example. Yeah. I think we are at the end, but this is really interesting. And, I wish Bernardo didn't have to go to another seminar so we could thank him, but let me thank you, Paola, and also thank you for participating in that committee, because I think the report was really, really fabulous. And I have to say that in response, Carolyn Montojo at Dana is trying to figure out whether a consortium of foundations could come up with some way of having sort of deliberative democracy around these kinds of controversial issues. It's certainly not easy in the United States. Part of it is because of both religious and political, very strong views on the part of some people. But every NAS report ends with, and we need further discussions, including the one on heritable germline editing. And of course, we have no venues or ways of having those public discussions. So to be continued, but thanks for that great report.
And thanks to you too, Steve. It was really interesting and educational for us all. And before the primer, you both, and with Matt Tetmeyer, came up with a list of readings, and I will circulate everyone by email for those who read more into this topic.
If you read them all, we'll give you an ersatz master's degree. And thanks so much for all of the people who don't normally attend this meeting, who visited and who commented. So thank you very much.
Really great. Thanks, everyone.
Thank you.